Cells need to maintain certain conditions to ensure that enzymes will perform their function properly. Each enzyme has an optimal or most favorable temperature and pH level. For example, in pH, gastric juice is made up of digestive enzymes such as pepsin, hydrochloric acid, and other substances that are important for absorbing nutrients. The enzyme pepsin is only active within the pH range of 3 to 5 and requires the acid to maintain that pH. Now let us guess the optimum pH for the enzymes in this graph. The peak represents the optimal or most favorable pH. For salivary amylase, the optimum pH is 7. For trypsin, the optimum pH is 9. Another example which involves temperature is the yeast. Yeast contains an enzyme catalase which helps break down hydrogen peroxide. Catalase functions best at around 37 degrees Celsius. As the temperature gets warmer or cooler than that, its ability to function will decrease. From the graph, we can see that at the start, the enzyme is still inactive, but as the temperature rises, there are more collisions between the enzyme and substrate, thus making more products or increasing the rate of reaction. The condition will reach a specific temperature wherein the enzymatic reaction occurs the fastest. This is the optimum temperature of the enzyme, which is 42 degrees Celsius. However, beyond this temperature, the enzyme starts to lose its ability to catalyze the reaction. This is mainly because the increase in temperature caused denaturation or some changes in the shape of the active site, making it non-functional anymore. Let us guess the optimum temperature for these enzymes. For the human enzyme, it's 37 degrees Celsius. For the heat-tolerant bacteria, it's 77 degrees Celsius. Now let us look what will happen if we increase the concentration of the substrate. Adding more substrate to an enzyme catalyzed reaction should also speed up the rate of the reaction. But look at the graph. What happened here? The graph shows that it reached a point that even if you add more substrate, the rate of reactions remains the same. This is the saturation point. How did this happen? If you add more substrate, it will increase the rate of reaction. However, this is true when the concentration of substrate is low. But above a certain concentration, all of the enzyme's active sites become occupied with substrate molecules, and the reaction reaches the saturation point. At this point, adding more substrate will not speed up the rate of reaction. Instead, more enzyme molecules are required for the reaction speed to increase further. Enzymatic reactions can also be regulated by the cell in the presence of specific molecules known as inhibitors. But let us recall first how a normal enzymatic reaction occurs.
During the normal enzyme catalytic cycle, the substrate encounters an enzyme with a specific active site to which it binds, forming an enzyme-substrate complex. The enzyme then facilitates the breakdown of the substrate to its products, which part from the enzyme, leaving the active site free to catalyze another substrate as the cycle begins again.
Now, what happens if there is a competitive inhibitor?
Competitive inhibition occurs when an enzyme encounters a blocker, which mimics the properties of the substrate and binds to the enzyme's active site. Thus, when the substrate is encountered, the active site is not available for attachment and no reaction will occur.
In competitive inhibition, The inhibitor molecules that are similar to the substrate bind to the enzyme's active site but do not react. Because the active site is occupied by the inhibitor, the enzyme cannot bind any substrates to catalyze the reactions. As a result, inhibitor molecules compete with the substrate molecules for the active site and overall reduce the rate of the reaction catalyzed by the enzyme. What will happen if the inhibitor is non-competitive?
Non-competitive enzyme inhibition involves the binding of a blocker to the enzyme away from the active site. This binding causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding. No reaction will occur as long as the non-competitive blocker is bound to the enzyme.
In non-competitive inhibition, it involves inhibitors that do not bind to the active site. Instead, these inhibitors bind to a site different from the active site, known as allosteric site. The binding of a non-competitive inhibitor to an enzyme changes the conformation of the enzyme so that the active site is no longer able to bind to the substrate. But why do you need to inhibit enzyme function? Since blocking an enzyme's activity can kill a pathogen or correct a metabolic imbalance, many drugs are enzyme inhibitors. Disulfiram or antabuse discourages drinking by inducing immediate unpleasantness. It is an aldehyde dehydrogenase inhibitor so that acetaldehyde, a toxic metabolite of alcohol, accumulates. Erythromycin acts by inhibition of protein synthesis by binding to the 20S ribosomal RNA molecule. The 50S subunit of ribosomes in susceptible bacterial organisms. Aside from digestion, enzymes catalyze all kinds of chemical reactions that are involving growth, blood coagulation, healing, diseases, breathing, reproduction, and many other biological activities. Photosynthesis and cell respiration, which you will learn more in the next two lessons, provide energy for the cell with the help of enzymes. Just as worker bees are important for the survival of a beehive, enzymes are the chemical workers in cells.